Okay, so let's just get back to it. So yesterday, this problem has been driving me crazy. We need to figure out what the overhangs are. So let's just make sure that I'm in the right spot. All right, so let's do it. Let's do this. 33 million. All right. So, okay. Um, this is our classic picture. We have a 5, a 3, we have a junction here. We have A, B, A, B, G, letter, string. And we can also do this. Say that this is so in the linear order, if we were to break here, we would get then we would get this goes into BRC becomes the 5 start and A becomes the 3. But no, that's correct. All right, so then we'll get something. So let's put this 5 here. Okay, then we'll have a 3 and a 5 ARC. This is just the RC. But so, and then if this whole thing is X, then this will be X. Then this should be XRC, and this should be B. Okay, so this was one double strand. Now, the thing that is driving me absolutely crazy is if we have this string this one that has the good picture. Actually, I think this one doesn't have a good picture. Oh yeah, here you go. But that's not even right. Wait, is that right? So we have this string. BRC is here. This is not right. The 5 goes from A to BRC. This is all backwards, dude. What the hell? That's not even right. Okay, this is right. So let's just draw that whole thing again because clearly I forgot. All right. Control dot. All right. Yes. So we're starting off with this picture. Let's just drag this. Okay. So we also have so this is a 5 and this is a 3. So in this one, we get another circle. This one, we have this break here, and we have a B and an A, R, C. And the 5 is the okay, so we should get so we should get so what happens? Our primers are A, B. So then A this goes into oh Jesus. No, but then if B binds to here, that's not right at all. because the idea is that I swear, man, I thought I understood this. Oh, wait. Okay, okay, in the process of this, I got totally mixed up with my conventions. Because my convention. Oy, oy, oy. Okay. All right, we need to start from scratch. That is insane. Okay, so what is still correct is that if you have wait, this is the old one. Oh, we want to change that. Okay. If you have, this setup here, these are the inverse primers. And normal. Okay. So that's still true. It's just um or I guess that's what am I trying to say? I'm trying to say so this is actually not right. Um Inverse primer locations. So what I'm trying to say is that the things that bind to A and B, i.e. the right, not so basically actually in this setup what we want is that ARC, so ARC here will have its 5 going 3. And that will be, so this is actually the inverse primer. So, this is all trash. Um okay. Okay. Well, that's actually fine. All right, so let's start over from scratch now that we know what's going on. So, let's just say that we have Uh, so why did I do it like this, and why did I decide to change it? That's, I guess, what I wanted to know. So we decided that we have this string, and it's because we wanted to call the inverse primers A and B that we did this. But let's confuse ourselves even more, and let's try to do this. So let's try it this way. So we have A and B on the top. This is new. I've never done it this way. Okay, let's say we have X here. So then we're going to have B, R, C, X. I'm just going to do star or dot. We're going to do a dot and a dot. Yeah. Dot. Is dot the best? Maybe bar. No, even bar is kind of dumb. Okay. So if we do it like this, let's just say we denature this thing. I don't want to do the dots, but this thing wraps around. And we have the 5. Let's do it like this. 5 going 3. No, so X is not actually here. This is not X. This is wrong. A is literally this sequence. B is literally this sequence. X is like this. And this is X bar. Okay, so what's going to happen is then A bar is going to bind to here with a 5 prime going 3 prime. And it will add bases all around so that we end up with a linear A bar prime, whatever it's called, X bar, B bar. Okay, then this is bound to this thing, but it denatures, so the loop, so it releases from the loop, and we're just left with this. But now this is the weird thing, right? Is then we should want a 5 going 3, B here. Oh, no, that's consistent with what we know. Okay, so then this is a primer, and this is a primer. So the primers are okay, let's draw the other circle just to make sure that we know what's happening. Okay, we have this break here. We have A bar, B bar, and the 5 going 3. Okay, then what do we want? We want B here. And then we'll get this string here, so this turns into 5, B. This should be X bar. Wait, is that right? No, because this is X bar, so this should just be X. Okay. And then what binds to this? We're going to get our A bar. So we get our A bar here. So this is 5, A bar, going 3. And then we have 2 strands. Now we have 2 copies, 2 double-stranded copies of the linearized DNA. Okay, so that actually clarifies my understanding a good amount. It's not that the primers are actually the top left and bottom right. It's that those are the locations that you want your primers to bind to if you're going to go the other way and have a junction in the middle. Okay. All right. All right, so now in this model, what we want to do is we want to write out the 2 strands. So the 2 strands are going to be 5 A bar, X bar, B bar, and then A, X, B. And you're allowed to do this thing where you like flip it like this, and then you like flip it like this, and then you can see that these are equivalent. So these are the same. So now what we're trying to figure out is, let's say we have this one. And we could actually draw it. Maybe we could draw it like this. So we'll have 5 going 3. And this will be A bar, X bar, like this. Well, we kind of need both strands. Okay, so let me draw it bigger actually then. Does undo propagate? No. Okay, maybe I'll just keep this so that I can erase from here. Okay, so if I draw a big circle here, like this, and like this. Okay, so let's make this the 5, the 3, the 3, the 5. So this is going to be A bar, X bar, B bar. The fuck? Okay. Then this one will be A, X, and B. Now, exonuclease is going to chew back this guy, and it's going to chew back this guy. So if we have our gene here, and what we want is that the... Hold on. Okay. All right, so now we're talking about the gene that we're trying to insert into here. And my expectation would be that the convention is that the outer strand is the one that we care about, and it's 5 going 3 like this. So then this one is... So then if that's 5 going 3, then this is going to be A, B. Is that right? And then this one would be 3 going 5 on the inside, and it would be A bar and B bar. And then so this turns into this. So this outer one has 5 going 3. Wait. What? No, I messed this up. This is A, this is B, this is A, this is B. Very, very confusing. Okay. But that's correct, I think. So, oh, you can't see it. So we have an A and a B on the outside. So A, to no, see that I was wrong. That's not right. That was wrong. Okay, I need to. Okay. All right. So I'm just trying to visualize basically where this gene goes, because in my mind we have this thing like this. Okay, and then the gene comes into there. And that's it. And the problem is figuring out that we need these overlaps and what the letter sequence is. Because when we think of the gene and we order a sequence, we order this 5' to 3' sequence. We don't give them anything besides just a string of letters. And it's implicitly assumed that we're giving them the 5' to 3'. So this is like this is really the gene, but of course they give it to you double-stranded. Okay, so if we did this and we had B 5 3 3 5, then B A B bar A bar, and the actual like promoter, coding sequence, etc. This is all in G, and then it's really just these overhangs that we're trying to figure out. And for the sake of simplicity, we've decided that we are going to keep the length of the primers and the overhangs the same so that we can use the same symbols for both. Okay. So what does this need to do? If this is, then this is a 3 and this is a 5. This thing gets chewed, and this thing is going to get chewed as well as this thing. Okay, so then that means that this needs to be A bar. So that means that this needs to be A, I think. Because then this thing slots in, and you have like a 3, 3, 5. And this part is an A. This is an A bar that's been chewed. And we're trying to add this to this, which is a 3. And this is a 5 that's been chewed back. So this part is A bar. So A bar and A will bind together. But the question is, so then we're left with I think that's a nick, and we have an A right here. Then we have a G right here. Okay, now okay, so what do we have now? Let's draw it as our big circle. So now we are going to end up with like this, and let's do it like this. Okay, so basically we had like this, oops. basically we had this A here, and we're gonna have this tiny little nick there, and then we're gonna have a G here, and then so this is still the 3' end. This is gonna be G bar, and it's a 5, and we just need to figure out what this part is, and this is an A bar. And so here what we have is a 5 and a 3, and this is a B, and this is a B bar. Okay. And well, basically what we want to know is if this B gets chewed back and this gets chewed back, then we want these to mate. And if these mate, then this needs to be a B. So then A, G, B is the gene sequence for the starting with the junction labeled as 5 A, B, 3, 3, 5, B bar, A bar. And inverse primers A bar and B, which we can see from, yes, A bar and B. Okay, so now the thing that's weird to me is like, is that the only it just seems confusing because you can totally put a gene on the other strand, right? Like, okay, let me download Snapgene now. I'll need to get my password. I don't have my password. I wish they had a sign in with Google thing. That's the best. Okay, let me get my phone. I was trying to avoid getting my phone today. But Snapgene, I gotta be honest, is pretty sweet. Okay. Okay, let's just type it. I don't even give a crap. I don't even give a freaking crap, bro. Okay, so let's download it. And hopefully there's no annoying... Yeah, there you go. Is this thing gonna work? Okay. Why isn't it opening? What the hell? There we go. Oh yeah, I need to log in again. Wait, what? Snapgene uses a font that is not installed on your computer. Yeah, let's try to reopen it then. Should be fine now. Okay. Um, open files. Did we get pull the bio thing? Okay, so here's Puck 19. So what I'm trying to figure out is like there's two strands, but it's like, does biology really, or is it just engineered that way? Like I know that we learned about this. Oh wait, do I need to? I don't know if I need to do this or not. Make sure Snapgene is closed, download and move this file to, okay, well that's hilarious. Okay, let's close this then. Move downloads / snapgene license .lick to .snapgene. Okay. I mean, that wasn't that hard. Okay, so I should be all activated. How much is Snapgene? I don't care. I don't care. All right, so oh, okay, wait. So if the arrow for a feature is running uh is going counterclockwise, it's usually that the feature is on the reverse or on the other strand. So if we're going 5-3 clockwise and we have 5-3 counterclockwise on the reverse strand. Okay, that makes sense actually. Okay. Ugh. Okay, so let's go back to the Okay, so B, X, A, then we have the G, and then we overhang this B so that when these get eaten up, I feel like it's right. I feel like AGB. Okay, so let's say we have the following setup for letter strings A, B, G, X of DNA. Okay. Uh, we have a plasmid, and we are looking at a junction where star represents RC. And we have 5 prime A or A junction B 3 prime and 3 prime A star. Let's add some spaces in here. Junction B star 5 prime. And we want to linearize it and then insert the letter sequence like a 5 prime B X A. Is that what I have? 5 prime B X A, 3 prime B star X star A star. The A the A star and the I want to write this down. Okay, cool. I mean, I think it's right. AGB in the for this setup. You know, we kind of been messing around with conventions and it's a subtle change. You know, like at the start of this I was like, "Wait, A and B are the primers?" And then I look at it and A and B are not the primers. It's the things that bind to what's going to go outward. So that's kind of the confusion part. Um okay. I kind of like this convention that you have just the A and B on the top as opposed to try to do it like on the diagonals. I guess we can add the Okay, so now we should be able to basically, so when we went to here, and then we went to actions, hi-fi, insert fragment. So this thing is really cool. So we're gonna linearize by PCR, and then we're gonna go here, grab the primers. This is gonna be the primer, and then this is gonna be the primer. And here it's gonna say this, but we want to reverse this and reverse this. So now we have this thing. So we're taking out lax ZA and the cap binding site and all of this other crap that we don't need. And so if we were to take, let's see, can we look at the sequence view of this thing? So, okay, so here we go. So we have this thing going this way. So this is, yeah, that sequence. Then if this is A, right? If this is A, then, and this is B. So yeah, so GACAGGTT, whatever, that's A, and then GAGGTT is B. Then we just need that to be in the gene sequence. Okay. Okay, so I think that we could just put in the direct thing of M Cherry, no promoter, and just see what the overhangs are. Um, Wait, let's actually do this then. So, file, new DNA file, enter a sequence. It's linear, and let's just say this is mcherry.dna. Okay, and then we let's just, all right, I guess that didn't work. Let's just, please import double-stranded DNA files. Okay, wait, can I choose? There we go. Nice. Use as a template for PCR. Use directly as the fragment. I don't know what that means, but I'm guessing that means, so it doesn't determine the primers for me. To choose the primers automatically in the vector tab, select the site of insertion to be replaced. In the fragment tab, select the region to be amplified. Amplified region, I want the whole thing. How do I do that? We need to watch that video again.
tutorial, I will show you how to simulate AnyBuilder hi-fi assembly with a single insert in Snapgene. To simulate the assembly with multiple fragments, check out our other tutorial. To begin with, go to Actions, AnyBuilder hi-fi DNA Assembly, and select Insert Fragment. The first step is to define and prepare your vector for assembly. If your desired vector file is already open in Snapgene before opening the AnyBuilder hi-fi assembly window, it will automatically be selected. To choose a different vector file, simply use the drop-down menu to pick a recently viewed file or specify another file located on your computer. The vector must be linearized for the assembly, either through PCR or restriction digestion. If you're linearizing your vector via PCR, select the Linearize by PCR option. Then shift-click on the two desired primers in your file. Snapgene will highlight the amplified region in the map view. If appropriate primers are not present on the vector, Snapgene can design them for you. In this case, select the vector region to be amplified, or simply click within the sequence to define the point of linearization by PCR. Alternatively, select the Linearize by Restriction Enzymes option if you're using restriction digestion to linearize the vector. To specify the restriction enzymes, you can either use the drop-down menus or shift-click on the two enzymes in the map view to select them. Finally, if you need to change the orientation of your linearized vector, use the arrow buttons here. With the vector sequence prepared, switch to the Fragment tab. There are three ways you can add your fragment sequence. You can use the drop-down menu to the right, or drag the sequence file into the window. Alternatively, you can simply select the Click Here option to find the file on your computer. With the fragment file selected, you next need to specify the sequence of interest. For this, choose a feature in the map view, or switch to Sequence View and click and drag on the desired sequence. Next, switch to the final tab called Product, and select the Choose Overlapping PCR Primers button to adjust your primer settings. In the new window, set your desired
To choose primers for the vector, select the site of insertion or the region to be replaced. I thought I did that. There we go. 40 overlapping. All right, let's just do 15 to 35. I don't know. That is pretty cool. We will walk through because I think that those primers, the yeah, we'll walk through exactly how the overhang method works too, because it's pretty clever. Very cool, man. Very cool. So we'll say this is Puck 19 M Cherry. No promoter. So I guess we need to just manually add the annotations. All right, so let's get into how well, I actually didn't like this thing because I guess okay, I guess what you're supposed to do is like we do this and then we annotate and we like do this and then I think I can right click, set DNA color, select all, select range. But how do I add an annotation? Features, add feature. Type misc feature. Oh wow. Oh, I hate that. I hate that. Hate that. But so what is the correct label here? Promoter. Is there just like a coding sequence thing? Oh, Gene. Maybe we'll just say Gene. So this will be M Cherry. And I just do OK. But then I want to edit it, right? So if I just double click on it, then I should be able to just say 1, 2, end. How do I say end? See, that's kind of dumb, but that's fine. Can I just click and drag it? No, I can't. So how long is this? It's 711. So let's just make it 700. Okay, so presumably if I save this, okay, yeah. So now we have the DNA file, and DNA file is actually a text format. So if we look at this, or okay, I guess it's not a text file. That's kind of random. Oh, okay. .dna is a snapgene file, but GenBank is a text file. Okay. fair enough. Okay, so we can try it again. Actions, hi-fi, insert fragment, vector. Okay, and actually what we probably want to do is we probably want to duplicate this thing. So puck 19, duplicate file. Okay, so then we rename this, and this will be with inverse primers. So I want to keep, because I want to keep the, and I don't know where this gets saved, but it's not getting saved in my sequence. Oh, it is. Nice. Puck 19 with inverse primers. So now we can go to the README. We can get this, and then we want to add, let's see here. So we want to add a feature to features, actions, oh, I should be able to do this. Okay, add primer. And I want the reverse complement of it. Okay. I don't know what any of these things are, but so this is going to be the I don't know what you're supposed to name them, but it's the inv primer 1. Oh wow, you can put okay, that's funny. Okay, sure. Add primer to template. Okay, cool. And then now we want to add another primer. Control R. And we want this to be this guy. And then and we want the reverse complement of it. And I guess this is in primer 2. Okay, so now we have 2 primers. And now what we can do is when we go to the actions, hi-fi, insert fragment, we can just select this primer and this primer. Oh, wait, no. This primer and this primer. And then, oh, great. So there we go. So now all we need to do is the M Cherry thing. All right, we're actually getting ready to order this thing. I'm actually kind of excited. We just need to now figure out the promoter, the operator, and the ribosome binding site, and then the terminator. We just need everything around M Cherry, but presumably I mean, I know that's going to be annoying still, but we are getting kind of close to this. So I don't actually know what polymerase for making blunt ends. Oh no, we don't want the A or G over. Okay, I know what that is. That's for like that weird what do they call it? Topoisomerase or something weird like that. So now we just want to select this feature, right? So we've selected a feature. NEB turbo. Oh, okay. Yeah, we do need to like, I don't know what this is, but apparently this is something for, this is like the generalized antibiotic thing or something. I don't know. Awesome. And I like that it's going counterclockwise. So, but we do need to inspect what. So we have. So what is this? Oh, wait, can we? Okay. Wait, can I save it? Okay. So, what is? So what is this? It's a 65 mur. So okay, so this is huge because then this is like 40. Okay, the overhang is 40, and then this is 25. This is huge, man. I don't think it needs to be that big, but okay, but the thing that we are curious about then is wait, how do we get back to that view? Right. Fragment 4, fragment rev, vector 4, vector rev. I don't know what that means. Okay, I'm guessing that this is the. Does it give me a description actually? What the hell? Um. Okay, so we have a directed graph here. So we have a fragment. Oh, 4 for forward. Okay, and fragment R for reverse. Okay, fair enough. So we got our forward primer and our reverse primer. And we amplify using them. So now we have an amplified. But the thing is, the fragment.4 and fragment.rev. Okay, these are the 65 mers. So then the result is a 791 fragment with the overhangs. Dude, this is so cool. Bro, this is so cool. Okay, so then hi-fi assembly inserts between, and then we have our new feature. Wow, pretty sick. Pretty sick. PCR with polymerase that creates blunt ends. That is what we want. Okay, so this looks pretty good to me. The only thing, all right, so we can just get back to business and fix the DNA sequence so that, and honestly, I feel like this thing about, oh, you need to use a, you know, a gene that's in the literature. I don't know that that's true. But one thing that is bothering me though is like if you go to add gene and you try to find something with tack or even with lack, but just like something so I can see like, cause like if we look at, all right, what does the amp R promoter look like? Can we get to that? So here, um, is this is 100? So the promoter here is 105 bases. And then I don't know why they've segmented it twice or into two parts. That's kind of strange. Okay, so wait. So let me add to the to-do list is to Okay, so we need to understand the. All right, so we'll just walk through this right now. Is understand the inverse or the overlap primers designed in Snapgene. Walk through arrow chasing of overhanging PCR. I think that's actually pretty straightforward. The arrow chasing for the overhanging PCR is basically the same as PCR. Let me get some coffee though. Okay, and finish designing the insert promoter operator terminator. We should probably actually try to understand bacterial transcription a little bit more in detail because, you know, to be honest, I don't understand how the terminator part works. I was sort of under the impression that there's just a stop codon, just like there's a start codon, but I feel like when we were reading the bacterial transcription Wikipedia page, it seemed like that's not actually what's happening. All right, so let's do some arrow chasing first to understand the general principle of how this works. Okay, so let's go over here. Let's go here. PCR with primer that overhang. Okay. So let's say we have a gene. It's linear. Okay. They denature. And we have a 5, a 3, a 3, and a 5. Okay, now let's say that we have, so we're trying to amplify this. We are trying to, the goal, let's specify the goal. We want, so let's call this G, want to amplify G, but also add a small letter sequence to each side. Okay, we want to add it to both sides, so that we're going to add basically a little piece, so that we have, let's call it like an A bar 5 3 5, and then this is like this, and this is like this. We add a B and a B bar, so we each, yeah, so A and B. Okay, so how are we going to do this? Well, let's imagine that we have a reverse primer here that binds like this. Okay, so this is P rev. And then we have another one here. And the punchline is that this is B and this is A, I think. It's either A and B are complemented, but it's, you know, something like that. So I kind of drew this kind of long, but A is like that, and B is going to be like that. So that means that this is a 5, this is a 3, this is a 5, and this is a 3, right? So what is RNA polymerase or DNA polymerase going to do? It's going to add to these bases up to here. Okay, same with this guy. It's going to come to here. So now what we're going to have is we're going to denature again. So we're still going to have this original template strand with nothing. So 5, 3. Let's call this C and D, so at least we can track the letters. So this is C and this is D. Then up here we have C and D. So nothing changed there. But now, on this guy, we got something new. We got a C, but we also have a B. We have a B. Okay? But now what's cool about this, and we're gonna ignore this one because it's gonna end up not mattering. I mean, I'll show you why it doesn't matter. But so now on the next cycle, what we're gonna do is, well, this is C. What does C bind to? So this is gonna be a D bar, and this is a C bar. So this is a D bar and C bar because of the denaturing. So, and same here. This is a D bar and this is a C bar. So what is this? So this is not quite right. We have a B, we have a D bar, and we have a C bar. Okay, that's good. But what is this sequence? Well, B, so D is made up of D bar and B, anyway, what is this? This is A. This is A. Yeah, that's A, I think. So then this is a C, right? So now, what is the primer going to do? The primer, oh God, I keep moving the fucking thing. This is like this. So now we're going to have a C here and an A here. But then what's going to happen? This is a 5. This is a 5, 3, 3. We're going to extend this way. But what's going to happen is that now, all right, let's denature one more time. So let's take this, denature. And now we actually have, this is going to split off into two things. One is just going to be the same old, or not same old, it's still bigger. It's C bar B with the D here. This is a 5, this is a 3. But what is this guy? This guy is amazing because it's what we wanted. It's A. So this is like, let's see, cycle 1, cycle 2, cycle 3. And this down here is a B. And then this is a C bar, D bar. And so now, we've basically extended A and B onto the ends of this thing. And just to show that it makes double strands that are kind of what we want. Because what did we want? We wanted C, B. Maybe I should have. Maybe I needed the complement. I think I messed this up to a complement. Because here we have A, C bar, but we probably wanted. Yeah, so we just wanted to complement these. So instead our primers would be what? A bar, or rather like A bar C. And this is a 5, 3. And then the other one is a 5 B, D, or B bar, D bar. Or wait, hold on. Yeah. Because then this is a... Wait, actually, hold on. So, oops. I didn't mean to do that. So, if I just click E, does it... Nice, okay. Let's see, these denature, we have this C here. We're gonna go... So this is a D and this is a B bar. So we actually wanted to make So we did want to make this a B bar, because then this is a B. And then we have... That's not a bar. That's something else. That's not right. So then we have A, C, D, B. So we get the C, D bar, B bar. We need to figure out what binds to this. This is going to be... Well, this is a 5. This is a 3. So this is going to be that A bar C. Oh, wait, no. This is just A. This needs to just be A. So I think it's... Oh, wait. Hold on. Might need to chase it again. C, D... So yeah, so it's definitely B bar, D bar. But then on this side, I think it's just AC. I think it's just AC. So AC. All right, so let's just take this top one. So the top one, it goes C, D, and then we ignore this. Ignore, because it doesn't really matter. Then this one, though, we get a C bar, D bar, B bar. And AC is going to bind to this and go this way. So we're going to have A, C, D, B. All right, here we go. A, C, D, B. Just like we wanted. All right, so we got it. That was it. Nice. So that's how PCR with overhangs overhangs works, and you still can add a little bit to it. I don't know the limitations of it. I don't know how long you can make it. I don't know, but for our case, we only need to add, I think it's like 25 to 40 NT overhangs for hi-fi. Okay, so we need to take, I think, the output of this, on the third cycle. So we're going to end up with, this is going to go into, and, but my question is basically, now we need to figure out how this goes into, this thing that we've been doing with the, what is this? This is supposed to be like... I believe that we want it to be like, I forget, we need to look it back over here. I mean, it depends, right? It depends on how we're labeling the junction and everything. So, okay, so, we need to go here. So fragment.rev is actually the primer that we... Well, that's interesting. Actually, it designed... Now, what is this? I don't understand that at all. Wait, inverse primer, I think I messed up, actually. I think that... Oh, no, I don't think I did. But I don't know, why is it 2 bases different? Why did it change it? Or 1 base different? $21, $23. So I don't know what exactly happened there. It's a little bit weird. Now let's go to the forward part of the insert. So the forward part of the insert is here. Or whatever I mean. I don't even know what I mean. So if we're calling this ATTCAT, the A part of our sequence, and this GAGG part, the B, then what did we compute that our overhangs were supposed to be? The overhangs are supposed to be just A and B. So then that means if the primer is adding to this, Wait, 5, 3, 3, 5. So that is gonna bind to the bottom part. So that's the lower. So this thing binds to the lower left, which means it's gonna go forward. I feel like I've been overthinking this the whole time. So the overhang really is just you add the overlap 5, 3, 5. So 3, 5. Pulls back my middle finger. Then we have our gene. This is 5. So this is A, because this is A. So then A complements on the bottom. So for real, it literally just is A and B. So if we have like an overhang of like this, for instance, then our gene sequence is just going to have that in it. The top. All right. Okay, so what's happening? Um Okay, so I think what we can do now is we can just get back to designing M. Cherry's DNA sequence. So, is there a naming convention? Let's figure out a naming convention. I am designing a gene that is basically P-TAC, LAC-O, M. Cherry, and a terminator. Because the thing about the ribosome binding site is like, doesn't everything need a ribosome binding site anyways? So, do you even need to specify? And I'm wondering what the naming convention is. Also, I gotta do this thing. Like, do I just call it P-TAC, LAC-O, M. Cherry? Motors prefixed with P. Operators name the variant and position relative to the transcription start site if it's extra or downstream. PTAC already includes a LACO overlapping the core promoter. If you add an extra... Oh, interesting. No, my strain has lac eye. I'm fine about that. I hate these papers, bro. They're so intellectual. They don't even tell you what the sequence is until, like, whatever. Like, I don't even understand. Why is this all separated out like this? I think I just want PTAC 1. I'm so sick of this ribosome binding site thing, man. I feel like it seems like as long as I get the spacing right and I pick the strong ones, it should work, right? Upstream terminator, PTAC, LACO, UTR. And then strong double terminator. Okay, so let me just write that down. So first we have P-tack with lack O operator. Then the second thing we have is this untranslated region and ribosome binding site. And then there's a spacer, right? Okay, so all right, so it seems like from the P-tack paper that there are two P-tacks. Where can I find a good official for my P-tack to copy into my cassette? It just seems like so crazy to me that like so much work is done on like, mammalian and all this plants and all this crazy stuff. But it's like we don't even have good compilers and stuff for just E. coli. It's like, why don't we just focus on E. coli and like make it easy to design and engineer E. coli? Even the Wikipedia article doesn't have the P-tack. Yeah. All right, let's read this. Let's read it. The tack promoter, abbreviated as P-tack or tack vector, is a synthetically produced DNA promoter, produced from the combination of promoters from the trip and lac operons. I actually don't know anything about trip. Oh, produced tryptophan. Interesting. Okay, it is commonly used for protein production in E. coli. Two hybrid promoters functional in E. coli were constructed. These hybrid promoters TAC1 and TAC2. Two hybrid promoters functional in E. coli were constructed. These hybrid promoters TAC1 and TAC2 were derived from sequences of the trip and lac UV5 promoters. In the first hybrid promoter TAC1, the DNA upstream of position -20 with respect to the transcriptional start site was derived from the trip promoter. The DNA downstream of position 20 was derived from the lac promoter. In the second promoter, the DNA upstream of was the DNA upstream of position minus 11 at the HPA site within the Pribnow box was derived from the trip promoter. The DNA downstream of position it also specifies a Shine-Dalgarno sequence, flanked by two unique restriction sites. The TAC1 and TAC2 promoters respectively direct transcription approximately 7 and 11 times 11 and 7 times more efficiently than the depressed parental lac UV5 promoter and approximately 3 and 2 times more efficiently than TRIP. Both hybrid promoters can be repressed by the lac repressor, and both can be de-repressed with isopropyl. Is this IPTG? IPTG, yeah, okay. This stuff is expensive, man. But you only need a little bit of it, but it's like, I don't want to spend $91 for a gram of it. That's so insane to me. It's like, dude, I don't know what the price of, drugs is, but drugs are cheaper than this stuff, man. Consequently, these hybrid promoters are useful for the controlled expression of foreign... Consequently, these hybrid promoters are useful for the controlled expression of foreign genes at high levels in E. coli. I mean, this is what I want. I want TAC. In contrast to the TRIP and LAC, LAC-I promoter not only has a consensus minus 35, but also a consensus Pribnow. Bro, this stuff, it just seems so arbitrary. This may explain the higher efficiency of this hybrid promoter with respect to either one of the parental promoters. Why don't they give me the C? Okay, so we want... Okay, so this was actually helpful because that means that we want PTAC1. So this is the sequence we want. And then ATG... That's methionine. That's... We don't even want that. So then... So this is everything. This has the AGGAAA... So that's the Shine-Dalgarno thing. But that looks like too many. AGGAAA. I thought it was supposed to be AGGAGG. Is it supposed to be AGGAGG? But alright, so we have this, so let's count. Let's do some counting. If I remember how to count. AGGAAA. 1, 2, 3, 4, 5, 6, 7, 8, 9. That seems like too many. This Echo R... I guess the reason why I hate this so much is that it is so specific to the geometry of RNA polymerase. RNAP is like binding to this thing, and these spacings, it's like you would assume we have the molecular dynamics figured out of you can just tune it how you want, but we're not there yet, I guess. Okay, let's keep going to Wikipedia again. The TAC promoter is used to control and increase the expression levels of a target gene and is used in the overexpression of recombinant proteins. The TAC promoter is named after the two promoters, TRIP and LAC. Bacteria promoters consist of two parts, the -35 and the -10. These two regions bind the sigma factor of RNAP, which then initiates transcription of the downstream gene. The TAC promoter consists of the -35 region of TRP and the -10 region of LAC. The TAC promoter is therefore inducible by IPTG, whilst allowing higher maximum gene expression than either TRP or LAC. This makes it suitable for high-efficiency protein production of a recombinant protein. The strong repression of expression in the off state is important since foreign proteins can be toxic to the cell. It's one of the most widely used expression systems, except it's impossible to find the fucking sequence for it. You know? It's like, man, biotech is out here in the Stone Ages with tooling. You know, I thought tooling in freaking software was bad. Talk about tooling in genome design. That's bad tooling. The TAC promoter finds various applications. One of the most widely used expressions is PTAC is a strong hybrid promoter composed of the -35 region of the TRP promoter. Dude, I'm reading the same thing over and over again, and they don't give you the actual sequence. The expression of PTAC is repressed by the LAC. The LAC-IQ allele is a promoter mutation that increases intracellular concentration of LAC-I repressor, resulting in the strong repression of PTAC. In addition, the inducer IPTG inactivates the LAC-I repressor. Thus, the amount of expression from PTAC is proportional . You know, in comparison, can we just look? I'm just curious. Okay, so lactose, on the other hand, per gram is like 20 cents a gram. So that's pretty good. I'd probably just plate my cells with, or I'd plate with lactose, because I'm not going to spend all this on IPTG, bro. IPTG is for suckers, man. I don't care if it gets metabolized. I don't want to spend that much on reagents. Wait, can we look at the paper again? So ATG is... Okay, so here we go. So this is that P-trip, and this is P-lac UV5. So methionine... But this is what I don't understand is, like, does RNA polymerase not care about the length of this thing? Because also this shine... But that's... I don't understand. They're saying the shine... Okay. DNA sequences of the TRP, LAC, UV5, TAC1, and TAC2 promoters. The -35 sequence and the PRIBNOW box sequence are underlined. Okay, so this is that 17 length spacer thing or something. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So it's like kind of... Okay. And so then this one should also be a 17, because this is the -35 region. And remember, the -35 and the -10 are from the middle to the middle of the 6-letter sequence to the +1. Oh, this is actually, now that I'm looking at this is starting to become clearer. Okay. Okay. Dots indicate every 10th nucleotide. Oh, I thought it was like a restriction site or something. Okay. The Shine-Dalgarno sequence in the ribosome binding site and the start codon of the HGH mRNA are overlined. So why is their Shine-Dalgarno sequence so shitty? Why would we not use the good one? Why would we not use... Yeah, why would we not use AGG? All these guys are using AGGAAA or something stupid. But everyone knows that this is what you're supposed to use. And actually, I don't even know why you would use this instead of TGG. Or wait, TGG AGG. TGG AGG? I don't know. This stuff just seems so arbitrary, but we just got to get it done. All right. Okay. The trip repressor binding site 13 and the lac operator 14 are indicated with broken lines. Okay, so this is the trip repressor site, and this is the lac operator site. The transcription start sites are indicated with +1. In the case of TAC1 and TAC2, it is assumed that the transcription start sites are in the same region as those of the lac UV5 promoter. So wait. The operator starts... Oh yeah, the operator starts at +1 because then transcription can't start. The transcription start sites are indicated with +1. In the case of TAC1 and TAC2, it's assumed that the transcription start sites are in the same region as the lac UV. The fusion points between trip and lac sequences are at the open spaces in the TAC1 and TAC2. Oh, okay. This is just their engineering. This is all done by the people at Genentech, right? Dude, that's so sick, man. Bro, the 80s of biotech is so sick to me. I don't know why, but it just got such a vibe, bro. Everyone else is writing software, and these guys are like, yeah, we're optimizing the fuck out of this promoter. It's just so cool that they've been doing this for so long. You know, I don't know how they did this, because it's like how could you do this without sequencing? Or without like good access to sequencing? It's insane. All right. So I don't know. Can we just use this and then change? The thing that I don't like is that, okay, wait, if I just copy and paste this, I'm sure that it'll be transcribed wrong. PTAC1. PTAC-I. Uh... Should I just use PTAC1? If I use PTAC1... I mean, okay. I could just use it. These guys are from Genentech. They're smart enough to know that it works. They're not going to publish this if it didn't work. So if that's the case, I could literally just sit down and write this out by hand. Except, what is this? I don't know what this is. GAGCTG? What is that? What is that? I mean, I could just throw it in, but I don't know what it does. I know what this is. I know what this thing is. It's like junk. I know what this is. I was thinking about this morning. It's like once we start to design like totally novel polymerases, it's like we'll be able to engineer this stuff that like there's none of this nonsense. There's none of this wasted spacing sequences. It's just gonna be like ching ching, and it just you know, it's like. So what is this? This is the okay. So if I look this up, this should be lac O. So if I put this into Snapgene, new Control N, lac operator, beautiful. I don't know what those extra lac operator Snapgene. So the lac operator is 17 base pairs, and I just copied in 17 base pairs, right? This is more than that. This is 21 base pairs. So they thought that the lac operator... What happens if we just do this? And that looks pretty good to me. But why is the... Okay, if we look at... Can we look at Puck 19? Okay, here's the lac promoter. So, between them... Okay, so this lac operator, is this the actual same lac operator as... TTGTG AG TTGTG AGCGG ATA ATA CAA. Okay, so that actually is the same. The lac operator is the same in PTAC as it is in Puck 19's lac Z promoter. So I'm wondering if we just like take this whole thing, copy it, because what is their see the Shine Delgarno sequence here is AGGAAA again, and then let's see how many spaces did they have between that AGGAAA. 1, 2, 3, 4, 5, 6. I guess it's 7. I don't really know how they count the off by ones. But I'm low-key, I want to take this thing, just copy it, then in this sequence, we go like this, except instead of this promoter. So wait, here's what I'll do. So I'll go back to Puck 19. I'll copy everything before ATG up to the -35 thing. So we copy this and close that. We can close that. We didn't need that because we have this one. Yeah, I'll save that. With okay. So, all right, so we copy this, and then at the beginning of it, what we're going to do is we are going to insert the we're just going to insert the thing from Snapchat. We're not even going to deal with this. So we're going to go back to, where did I see the TAC promoter? We're literally just going to copy this thing. Oh, that's cool that they have the thing with the copy paste. I like that they do that. It's smart, you know. It's like it's kind of Wolfram-esque, you know, context aware copying. Okay, so now we just add the mcherry text file, and the mcherry text file starts with ATG, so we're good there. So we should be able to just paste this in. And I wish I could like maybe what I should do actually is if I open up, let's open up folder like that. Nice, that's good. So we have mcherry.dna. And I said it was a gene, right? So then can I have it show me the... Is it possible to show me the... If I say it's a gene, can't it tell me the freaking amino acid? Oh, there we go. Okay, so we're going to copy this whole thing. And we're going to paste it into here. Oh, that's so annoying. Is there not a better way to do this? Oh, shit. And then I kind of want to do this. Oh, I hate that. That's the dumbest part about this thing, is that you can't just... Maybe that's just a bug in No, I think I experienced that yesterday. So we have the TAC promoter. Wait, that doesn't seem right though. What did we do in 19? Yeah, it's short. It's too short. But I don't know what we fill in the interim. So we have like the minus 35 thing, which is 6 BP. Then we have what, 17 BP of spacer, 18 BP of spacer. Oh, that's supposed to be 17. In the tack promoter, is it 17 or 16? This one's 16. God fucking damn it. Okay, fine. It's 16. This is tack. And then we have the minus 10, which is also called the Prinbo box or something. And this is 6 base pairs. Okay, then we have, so this is all called the tack promoter. And then we have seven base pairs. Spacer. And then we have the lack operator. And lack O is 21 base pairs, I believe. No, it's 17 base pairs. Simulate agarose gel? That's cool. Insert codons. Genetic code. E. coli. Where's E. coli? Oh, okay. I can't just paste in an amino acid sequence? That doesn't seem right. Insert feature. Does it have a search? Ooh. Okay, so this is kind of what I was... Okay. This is kind of... Now we're getting somewhere. So I should be able to just go to... What is it called? It's probably just called tack. Is there a search? Can I search? There we go. Tack. So we insert tack. And then what do we need? We need a spacer. Do you have a video? Where's their video? They don't have a video? Come on. Set the topology. Insert sequence elements. Insert bases. Manually type the bases. All right, let's just copy it then. Okay, so we have this. What is that? Insert bases. Then insert feature. This is going to be the lack. What the hell? Lack. Okay, so wait. Oh my god. Okay, so there's a bunch of different lacks. So that's the lack promoter. This is like a longer. So which lack operator is more common? Which should I pick? I think that this is the 17 one. So let's just do the 17 one then. So basically, we want to count from here to here is 34. That's kind of why I'm a little bit... Because shouldn't it be 35? Or... Okay. And then after the lack operator, what do we need? The lack operator, between the end of the lack operator and the start of the coding sequence for Puck 19 is 19 base pairs. Then we have 19 base pairs. This includes the RBS. All right, so wait, can we read the ribosome binding site? A ribosome binding site is a sequence of nucleotides upstream of the start codon of an mRNA transcript that is responsible for the recruitment of a ribosome during the initiation of translation. Mostly, RBS refers to bacterial sequences. Ribosome recruitment. Okay. The RBS in prokaryotes is a region upstream of the start codon. This region of the mRNA has the consensus sequence AGG. The complementary CCU is called the anti. Oh. So empirical. Bro, give me a break, dude. Kozak is the eukaryotic or what? Yeah. But it doesn't even have a map? But it doesn't have a gene or ribosome start thing. That's what it's like. Why would you even publish this? So where is that from? We describe a simple system for reversible, stable integration of plasmid-borne genes into the... Oh, whoa. Into the E. coli chromosome. I actually do want to know how to do that. That's part of my tech tree, bro. How do you get it into the host? And how do you test that you actually got it into the host? Bro, I love this guy. Oh. Early cloning experience may be conducted using usually. Man, we really need to look at the E. coli genome, bro. Okay, I feel like I'm procrastinating. Let's get back to this construct, though. I mean, no matter what I do, there's going to be some element of risk here. And I just don't... Okay, let's... Bruh. Search by sequence. Actually, let's do that. That sounds good. Oh. Oh, that looks pretty good, actually. That kind of looks like what I was hoping for. Snap tag. Okay, all we really care about is... So look, it's got a publication. Let's look at the sequence of this thing. Let's download the snap gene. Oh, bro, I like this, man. I like it. All right, so we want to go here. Sequence. Okay, so I feel like maybe we should just do this. So how many is that? That's 75 base pairs. Let's get rid of this thing. AA. AGG. Wait. Cagtat. I feel like we're getting ready to order this thing. Hmm. Hmm. Well, I can't believe I'm actually about to order this. Wait, what? It's pre-optimization. I feel like they don't even give you the fucking tokenizer. What if I add spaces in between these guys? Okay. Wait. This is the wrong freaking one. What we want to do is we want to actually do in our construct here. So if I do this, everything has its own token now. I think that this will work. I think I could order this. Bro, I think we should just finalize. I feel like this is it. So now all we have to do is add the overhangs. I don't know what this will be. This one will be 800. And I don't know what it will be. Bro, I don't know what it will be. We can make it like, let's just say 20. Let's just say 20. Ooh, I like this thing. But this does not help me figure out the overhangs. I think this might be what I want. Input source sequence. GenBank. I think I have the Puck 19 GenBank. Oh, I guess I can take the let's do this one then. Okay, fine. Wait, what? Wait, what? Okay, so let's just do this, and we're going to save this, and this is going to be the okay. M-Cherry 2 no overhang. I do like these tools, man. But this didn't... These didn't do any of the overlap, though. Yeah, so this doesn't work. Okay, so instead, let's go back to build, and then let's view overlap detail. Oh, good. Okay, so if we take this, this is A, and we edit this thing. So we're going to put this right here, and then can I go back? Oh. Oh no. Let's update this. This is a 5, this is a 3. 5, 5. I don't understand. This one seems right. Join is discontinuous. This is going to get eaten back, right? And this is going to get eaten back, right? So then it should anneal. So instead, if I added the other one, I could have sworn that it was supposed to be A and B on the other sides. Okay, so what do we have here? We have A bar. A is equal to 5 prime TGGA, 3 prime, and then this is equal to 5 prime TCCA. Okay. And we have 5. Oh wait. This is the 5. Yeah, that's the 5. This is the 3. This is the 3. This is the 5. This is A here. And then we have this synthetic fragment with a 5 and a 3. I feel like it should be that, though. I feel like it should be A is here. Done. Can I save this? So I think that was right though, but I think okay, so it was right, right? Because I didn't see any warnings on that thing. Load save. Something. Puck P tac Puck 19 tac P tac M cherry. Cool. So we should try. Well, okay, wait. Wait, hold on. Oh, this is the old one. Okay, so instead, if we I can't tell if this is good or bad. I think what we want to do is we want to change this. The forward primer. This is going to be in this one, right? Where did we do the actual? I think it's this. So that. So we want this is the forward primer. Forward primer annealing site not found. Will it do the? What? Oh, did I change this? That seems weird. Is the first thing not an ATG? Oh, you know what I need to do actually? Wait, hold on. If I go back, can I mute this one? Oh no, but this is the promoter. This is the promoter. This isn't even gonna have it anyway. now we just want I think we want this. Did I export this thing? I did. Okay. Mm-hmm. Five, three. Yeah, that primer doesn't seem right. Shouldn't it be the other way around? This sequence. Okay. No, that's all good. That's good. That'll work. So we just want to add this to our construct, right? So if we go here, P-Tech M Cherry 2 no overhang. Okay, yeah. Now what I want to do is do actions, design synthetic construct, insert. I want to insert. Do I? What about my features? Okay, all right, fuck it. So we're gonna go to sequence here, copy this. Go in here, paste this. Okay. So we have this. Now all we need is our overhangs. And our overhangs are going to be this thing. And this, right? So that's a 5 with B. So this is a 3. This is B. It's a 5. Then we have B prime 5, 3. This gets chewed. Now we have our gene here. This is a 3, this is a 5, this is a 3, this is a 5. This gets chewed as well. Okay, so this needs to be B. Alright, so that's it. That's freaking it. That's the whole thing, man. And it's saying don't even do the full 22. So I guess it computed that. So we did no spacers, because we don't need spacers. So that's good. Alright, so now we just add this at the end, and we're ready to order a gene from freaking... Okay, so now what I want to do is I want to file, save as, overhangs. Okay, so now what I want to do is I want to do another one of these things, NEB builder. New fragment, load file, select file, puck 19, okay, specify custom primers, puck 19, so this is the reverse primer. Wait. Oh, okay, and we need to export this DNA as GenBank. Wait, is that the right one? No, fuck, that's not the right one. Oh, you know what I think I messed up actually? Okay, we need to add this. But here, did it knew it was circular. Okay, good. It parsed the GenBank. So now if I click this, it says insufficient overlap to expect ends to join. But if I do this, TTG. Wait, what happened? We have the gag at the end, which is this is all kind of sus, but that's okay. The TTG. What happened? Where's the one that designed the primers for us? I thought I was supposed to, oh, did I just add the wrong part? Yeah, I'm supposed to add this. Wait. No, I never, oh shit. Oh boy. Oh, but this isn't even the, oh, it is. It is. Okay, wait. So this is, okay, so I don't know what happened. We want to save this, export DNA, save, overwrite. Okay, let's try that again. So we're going to delete, oh wait, no. Cancel. Delete this, add a fragment, load file. Did I re-export it though? I don't think I re-exported it. Did I? I'll do it again. I can't remember. Okay. Synthesis. We're getting better at this stuff, man. All right, so let's look at the that looks good to me, man. That looks good to me. So then I should be able to just buy this, right? This is it. Is this it? So nerve-wracking. Is this the reverse one? I don't even know what that is. Okay, so all right, we're gonna do this whole thing again in benchling because I feel like the more tools I do this in, the better I'll feel about it. Okay, so this is puck 19. We want to duplicate this, right? Copy to. Yes. So this is going to be called copy of puck 19. We can rename this. Okay. Now we can basically, let's see, how do we insert primers into this thing? Features. Okay, primers. Create primers. Pairs. Attach existing. No. Oh, wait. I don't think I wanted to do that. Okay, primers. Create primers. Manual wizard. No. Primers. Create primers. Manual. Okay. So the forward strand is going to be here. No promoter. I think it's supposed to be called no. Oh, no promoter. Yeah, that's right. All right. So here's the reverse primer. Did it find it? No, wait. Primer pair. Okay, this is the reverse primer. And then the forward primer is this. This is going to be inverse primers. Linearization. Save primer pair. What's wrong? Save to. Wait, did that save both of them or just one of them? Okay, so this should be a 2000. Okay, something is wrong. We didn't do it right. We're gonna go to puck 19. Wait. This is the forward primer. This is the reverse primer. Oh, these are the two names? Okay. In forward, in rev. Okay. That was silly. But also, I think that this is... Like, maybe this isn't even right. If we try to search in here? This is where I want it. Three prime location? I don't get what that means. There's only one in here. I don't get what the hell they think they're talking about. Like, this isn't even the right spot. So why are they doing that? There's only one of them. And it's right there. I think that's what I'll do. So this, I'll just do it like this. This is an easier way to do it. So this is the forward primer at one, at, what is this? Do we have the numbers? This is the forward primer. I think this is a different puck 19. I think it's different. See, why is that saying it's at 11? Oh, because this has the M cherry in it. I get it. So what we want to do is make a puck 19 with inverse primers. That's what we want. We just want that. There we go. Okay, so sequence, this is what we want. So we have this primer, which is the, okay, and then this one should be at 1060 or 1052. Perfect. Okay. So we're going to take this, create primer, forward primer. Let's make a pair. And then the reverse primer is going to be this. Oh, in selection. Stop doing that. There we go. But like, why is it putting it there? That's so retarded. What does that mean? All right. I think it's, hold on. I think what I want to do is not do this primer. I think this primer pair thing is just bugged. Save primer. So this is going to be in forward. Save primer. Okay, now we go here. We look for this one. We click on that. Create primer reverse. In reverse. Save primer. Okay, cool. So now we should be able to do create PCR product, right? No linked primers. Okay, how do I... Switch to the pairs tab and click on link primers. Oh, did I miss that? Primers, pairs, link. What? What did... Oh, okay. So now I have a primer pair. So now I can create PCR product. Please be 2,000 bases long. Nice. Okay, so we did it. All right, cool. Do they have assembly though? CRISPR. What is this? Alignments? No. Let me get some water. Learning these tools is definitely a bit of a struggle. So wait, can we do Gibson hi-fi assembly benchling? Do they have a thing? Oh, they do have it. So create assembly. By cloning, by concatenation. I don't even know what the difference is. Ugh. That seems pretty cool. Let's just try to use it. Assembly. But wait, which one do I need to pick? Cloning or concatenation? Ugh. Damn it. Okay, I need to get some water. All right. Um, assembly by cloning by concatenation. Okay, cloning. We're doing cloning. This tool is pretty good. So this is the synthetic one. This is the synthetic one that this one worked. And then this is the one by PCR. I like that diagram. That's a good diagram for PCR. 2 arrows. 2 arrows. Wait, what's the difference between okay, I think we want to do homology. And the number of fragment bins 2. And then this is going to be P-TAC or Puck 19 TAC M Cherry. Okay, so the backbone is going to be Puck 19 inverse primers. Wait, did it do it? What the hell? hate this thing, dude. There we go. Okay, yeah, we want the whole thing with the forward orientation. What is that? What is that? That's not right. Wait, why are these primers wrong? What the fuck? I think that was the bad one. That was the bad one. So we don't want that one. We want is it okay, so let's just delete this. Delete it. Archive. Okay, this one. Delete archive that too. That's total trash. This is also trash, but I feel like that might break something. I kind of don't like that they store these sequences on their own, but whatever. Okay, so copy of Puck 19. Here we go. Puck 19 inverse primers. There we go. All right, good. And then we verified that that this is the PCR product. So this is the PUC 19 Inv Primer PCR product. Good. So now what we want to do is go back to this thing. What is that? I don't even know what that is. That might be M Cherry. Here's our assembly. There we go. All right, so backbone, open sequences, Puck 19 Inv Primer. Okay, there we go. That looks pretty good. Okay, the fragment, let's just do plus entry DNA sequence, new import DNA sequences. S Glock? Bro. Snap Gene got a Glock. Holy hell. So what we're looking for here is PTAC M Cherry overhangs. I think we do want the overhangs one because it's going to automatically detect. Okay, so this actually looks pretty good. Methionine start. You know, I don't know about all this. This is a little weird, but presumably this is good. And then source. Source is like a feature that they imported that's kind of... I mean, I get it. I get it. All right, so let's go to the assembly. Insert. Open sequences. PTAC M Cherry over. More than one overlap was detected? Yeah, shouldn't there be more than one overlap? I wonder actually if we tried to do the PCR product instead. And we... Bro, what? I don't like benchling, honestly. Benchling is kind of annoying. Okay, how do I delete this one? I want to delete this guy. Okay, let's try that again. That looks pretty dang good, actually. And what is this? What is this orange? I don't know what that orange is, but that looks pretty good to me. The only thing is it linear? Is that it? I think that's it. Assemble. Select a schema. No schemas found. Next. Okay, so we should have a plasmid, right? That was actually pretty easy. I like it. I like it. That was cool. It detected the overhangs, and it nothing complained at me once I did the linear one. Ugh. Cool. All right, so um, let's just walk through the entire assembly procedure again, because I need to read about restriction digests, and I need to read about PCR cleanup, and for 50 PCR cleanups, I might do this. I honestly might do this. This comes with Okay, so is it all right, so I think we have the whole thing done. It's going to be the insert is at P. Whoa, what is that? Is that from Julia? That's ridiculous. um, seeks. It should be P-Tac M Cherry overhangs dot DNA. Okay, so let me just write this down. So my understanding of what is going on is going to be we have liquid culture. I know I've done this 1,000 times, but the repetition is helping me feel sane. Of E. coli with Puck 19. We are going to mini prep it. Mini prep, which gives us to get 100 nanograms per microliter of Puck 19 purified. We then are going to dilute that. Dilute it for PCR. And let me see, what did we have written? Because we already had a lot of this written. Okay, then we're going to add primers, master mix, or let's say inverse primers, master mix, and like, what is it? Like 1 nanogram or something? Okay, so can we run the direct result of PCR on a gel? I think we can. This is way more expensive than Monarch. New England Biolabs has got such a vibe, dude. Okay, we'll need to figure this out. But I'm down to like kind of go down the rabbit hole of making sure that it's pretty clean. I'll probably buy just the normal one. I don't think I need Precision engineered spin columns. We're not going to have 100 micrograms. 100 micrograms is 0.1 milligrams. That's a lot. 0.1 milligrams is a lot, dude. If you, I mean, it's not, it's actually not that much, but it's a lot of DNA. 0.1 milligrams of DNA is a ton of DNA. Okay, so, so 5, we're going to PCR it. And what we should end up with is, okay, so the PCR product, can we directly run So, okay, I think what I want to do is, in no particular order, we're going to need to order the Monarch PCR cleanup kit. We are going to need to, we're going to need to buy agarose. Okay, oh yeah, we need the ladder. I have some ladder, but I probably do want to buy the ladder. But we'll try to... Okay, do they have a PCR kit? But I think we want the good stuff, right? I feel like, Okay, um, so my plan was PCR, then 6 is run on a gel to see if we have um a 2 kbp band. Why do they call it a 1 kb if it reads from a... Oh, I guess it's centered around... Okay, cool. So this ladder is the quick load purple. N0550. N0550. Okay. No SDS. Oh, wait, actually?
When I was a young kid, I was really interested in genetics. Well, I didn't really understand genetics. I kind of thought that when two organisms had a baby, the baby was just this blend of the two. Yeah, that's a misconception. But I really saw genetics in action with my guppies. Guppies are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which means there are no eggs, like many other fish. And second, they have a lot of babies. They also eat the babies, but I don't think that's especially cool. So as you can see, that is not part of the cool fact list. Anyway, when my surviving baby
Bro, I love these guys.
they can have all kinds of cool traits. These traits were carried by their DNA, their genetic material, which is found in their body cells. But sometimes I would forget which mother was the mother of the baby guppy, because there were several mother fish in the tank, and I wanted to keep track of inheritance in my guppy notebook. So what would have been very cool to have at that time? Some biotechnology. Biotechnology is the merge of biology and tech, and it's constantly changing. It includes topics such as PCR, cloning, and genetic engineering. It's also an awesome field. We're going to talk about one of the biotechnologies that could have, well, potentially, helped me determine the genetic relationships of my guppies. If, as a young kid, I had access to it. Although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate molecules based on how big they are, their size. and it's especially useful with DNA. Let's look at DNA real quick. So here's the guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell. And if you were to zoom into the DNA, here is a nucleotide, which is a building block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis, which again separates molecules based on size, relies on the fact that DNA molecules have a negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight. They take more time to make it across the gel when you compare it to shorter DNA pieces, which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for
Why do you need to stain the gel? Loading dye just helps you pipette and track the run. Ah, okay. All right, I'm going to write that down. So, loading dye is only to track the front of the band. Okay. So, what's the general PC or electrophoresis setup? All right, PCR product. Okay, see, we're really starting now to like break down what is going to work and what's not. So, for instance, if we run it on the if no band, return to one and use new primers or something, primers or reassess. Um, all right. So if there is a 2 kb band, okay, wait, let's actually is TAE the cheap stuff? Wait, is TAE and TE buffer the same? Cause I can't remember. If we did TE buffer, I'm pretty sure TE buffer was cheap, but what about TAE buffer? Okay, it's definitely not the same, but it's also pretty cheap. Okay. No, but they're both. Okay, sounds good. Loading die. Unless your PCR mix is quick load. Um. Okay, so I need to get some. So wait, so here's the thing though, is the now let's close all this crap because who cares about that? The thing that I want to know is in so this has Okay, so if because I kind of don't want to use the ethidium bromide. I think I need to get the other stuff. I think I need to get the KB plus DNA ladder for safe stains. So do they sell their own? Markers and ladders? I don't know if they sell it. They probably sell it. Bro, this time it's Nebraska State Bank. That's ridiculous. So maybe we'll just get the 90. Because high affinity nucleic acid binding sites can affect DNA migration during electrophoresis, post staining of gels was. Wait, post staining? Okay, wait, what if we look at this thing and look at the protocols? Protocol for loading. This is going to be so dumb. Okay, 1 KB plus letters is stable for at least 6 months for long-term storage. Okay, that's fine. Mix well after thawing. Okay, all right. The 1 KB. Uh-huh. As a post-electrophoresis stain. So wait, what's the difference between post and pre? Post stain. So what? Wait, what is that stuff called? With the DNA stain. With safe stains, post-staining is recommended because pre-casting those dyes in the gel can alter DNA migration, make bladders look faint or smeary. After the run, let the gel soak in 1x CyberSafe. Dilute 10,000x stock in TAE. Oh, wow. Okay, fine. So I can still precast if I just need to verify that there is a 2 KB band, but no, but not going to do any further extraction. Because basically the plan is to take a little bit of the PCR result, run it on a gel, check that there's a 2 KB band, and if there is, we can then proceed with cleanup. Okay, so because it seems like just precasting the dye is way better. Wait, load 5 to 10 microliters of PCR product? I feel like that's going to be okay, that's fine. Plus okay, so we need to get this so we don't want this ladder, we want the other ladders. And I think I am just going to buy it. And then what's the storage here? And how long is it going to last? For long-term storage, store at 4C or -20. The thing is, are freeze-thaw cycles going to be bad for this? Because I could see freeze-thaw being a issue. The only thing is I just don't really have a space in the 4C for... Okay, well, we want to look up... Do they sell stain? I don't think that they actually sell the stain. Okay. Okay, so do not freeze, protect from light. Damn, bro, this stuff is so expensive. Wait, do not freeze. So then what am I supposed to do with it? See, that's insane, though. I'm not going to use 400 microliters of this stuff. Oh my god, these protocols are ridiculous, dude. In 6 months? Well, that's pretty neat. Actually, why? This responds to blue light way more than it does to UV. Do not freeze. What's a break box? That's dumb. That's the dumbest. I thought it was gonna be cool. That was stupid as. That was dumb. Um. Store at room temperature? Really? In the original container? What about the gel green stuff? Oh, Biotium? Dude, that's crazy. I actually was just in Berkeley and I saw someone with a Biotium shirt and I was like, what is that? 10,000X in DSM. I don't know what the difference is. Trial size? See, actually, trial size sounds pretty good to me. But what is this? This is gel red. And, okay. What about storage? Are they in Fremont? It's cool. The thing is, wait, actually, could I get a free sample? How do I get free stuff? Because that's too much, dude. 400 for something that lasts only 6 months. Okay. Okay, let's keep going though. So, all right, so if there is a 2 kb band, then we are going to go to, okay, so let's say we see a 2 kbp band. That means that we did the linearization correctly. Now we want to do PCR cleanup and DNP digestion. What is the correct order to do these, and what is the protocol? I have mini prep kit from Bioneer K311 kit. I don't know, do I need the what if that will work to purify my linear plasmid? Because then I can actually quantify it on the nano drop, which would be cool. I'm not personally convinced that DNPI or whatever it's called. Okay, so then and that was like this is really cool. Do they do a promotional video on this thing? Dude, NEB just seems like a dope company. Yeah, so PCR doesn't methylate the DNA, so that's why if you see this methyl group pronunciation, DPN1. Okay. Schizomers. Okay, wait, how much was this stuff, though? Fuck. God, fuck. $77. Okay. Okay, so we're nearing the point where we actually have a bill of materials, and then we can, like, go from there. I think a lot of it we already have in the lab, but, you know, it's one of those things that's like, you know. This was crying out for alarm. Okay, so we have, yeah. PCR 50 microliters. What the fuck is this? There's always something else, dude. What is Cut Smart Buffer? Oh, it's an NEB product? Lol. Oh, that's fine. Okay. This is only $29. But how long does it last? That's kind of my main thing. GMP grade reagent. I want to know more about that. Oh, interesting. Dude, do they have... What the hell? NEB New England Biolabs Tour. Oh, fuck. Look at that.
Close collaboration with our customers, really with an eye towards the future. New England Biolabs is not afraid to invest. If you look at the life sciences industry, New England Biolabs has been at the center of or behind a number of those tools that have enabled scientists to make incredible discoveries and breakthroughs over the last 50 years.
Slow motion. Get a slow motion.
On top of how products are being applied, production processes, and the technologies involved with making our products using the most advanced analytical capabilities. We're ISO 9001 certified and ISO 1345 certified. Our GMP grade line of products and the facility that we're sitting in today that was built to produce them are designed to be the highest grade materials that we can supply to the marketplace.
The manufacturing process starts with process development, ensuring the best fermentation and purification for our strains. Then the formulation group does functional testing and dilution down to the final concentration, ready for our filling and shipping groups to complete the kits to distribute throughout the world. Our goal is always to enable a customer so that they can do the research that they need to do. We can support extremely small volumes. We can support extremely large volumes.
We were an integral part of the ability of our customers to supply vaccine for the pandemic on a global level.
We really have the ultimate level of flexibility.
Bro, NEB is so
Scientists work with
badass.
A lot of our customers at very, very early stages in their development. We're supporting them in process development right through to preclinical and then into clinical and commercial production. The full life cycle of the product. Pretty remarkable. New England Biolab has a very broad portfolio of products, 1,400 SKUs in the catalog, and we serve different types of customers, academics, companies that make diagnostics, therapeutics.
We're very transparent as well with our customers, so they know when they buy a product from us that they're going to have full visibility, full traceability to how we make, test, and release that product to them. Really, everything is meeting defined expectations.
The consistency here is amazing.
It's important that we're able to get the product to the customer in the format that they request.
To help them integrate them in.
Bro, biomanufacturing is just the freaking coolest industry. I swear it's like, I don't know if I said this on stream, but it's like, if you think about lyophilized DNA pellets, okay? That is, just developing the technology to enable shipping DNA in a way that is stable is a massive breakthrough. So there's like a whole layer of like huge logistical things that you can do to make bio research go faster that's outside of the lab, which I find really interesting. So because it's like, if you think about the next, 10 years of bio research, it's going to be, I don't think it's going to be overnight that it's like really cloud labs and all this stuff that I dream of. It's still going to be sitting at a bench for a ton of people, and being able to design, because I guess what I'm trying to say is the places where you'll probably see a lot of automation first are these high throughput things where you're like manufacturing reagents and stuff like that. Because that's kind of like precursors to doing a lot more specialized stuff. So while a cloud lab that does, assays, cell culture, all of this assembly, all of these more like specific protocols, like that's killer, but it does make sense why it's been so slow. Like, it makes sense that like NEB, a place that does a lot of the manufacturing of stuff, is way more automated because they're producing such high volumes.
We've also expanded in the UK and added lyophilization capabilities.
By freeze drying.
They just said that. That's what I literally just said that. That's so funny because it's like, that is such a big deal.
It's really going to help shorten the development for our customers. Get a product to market. Adding lyophilization, adding this GMP-free facility continues to allow us to meet our customers' needs as they evolve and become more diversified themselves.
Let's fucking go! It's just like, what? Bro, what? That only has 300, because that video is kind of a nothing thing. Oh, they have a podcast, dude. That's pretty sick. Honestly, that video kind of sucked, though. There are a lot of good videos, though. What is their most popular video? Ooh, actually, I want to see this. This world is crying out for love.
Transformation is the process by which DNA is introduced into bacteria. It is thought that chemical transformation, which requires chemically competent cells, uses divalent cations to increase the permeability of the bacterium cell wall, thereby increasing the likelihood of DNA acquisition. Many common protocols include a heat shock step to improve DNA uptake. Electroporation involves the application of an electrical field to the competent cells in suspension, thereby introducing holes in the cell's perimeter. The newly compromised cell wall allows exogenous DNA to pass into the cytosol of the bacterium. Outgrowth of bacteria following transformation allows for repair and recovery of the cells in liquid medium without antibiotic. After outgrowth, the culture is spread on selected plates to isolate transformants containing the newly acquired DNA.
All right, this is boring, dude. It's like so obvious. Okay, so let's get back to the bill of materials. All right, so I'll probably get the Monarch kit. I'll probably skip the Agaros. I'll probably buy the ladder. But the thing is, what's the shelf life of the ladder? Because the main thing is like buying this. I just don't use that much of it. Storage. So basically what we want to do is we want to create a spreadsheet, I guess. And we want a spreadsheet of the bomb that's like you know, the SKU, the name, the storage, the volume. Two years? 2 years. Who knows what's gonna happen in 2 years? Oh, now they're linking me to Nebraska.edu. This is legitimately like the best freaking meme, dude. Oh man, that's so funny. I always forget the name of this. What is the name of that job that deals with like sourcing parts and like finding out how to get them and acquiring like stuff. Procurement, bro. I always forget the name procurement. But yeah, procurement. That's what we're doing now. Okay, so that's good. And that's on the product spec. Okay. So wait, is there's a full document that comes with all the NEB stuff. Let's close all this extra shit. There's a lot here. We can close all this because for the most part we did it all, right? We did it. So here we go. So this is the thing we're buying: protocols, manuals, and usage. Quality, safety, and legal. Literature, tools, and resources. Where's the official product specification? So they actually recommend storing it at 4C. Dude, I would love to talk to someone about how they do stuff like this. Maybe just ask ChatGPT. But oh wow, here we go. Nice. Okay, so it's under quality, safety, and legal. That's great. Okay, so look for find the NEB. All right. Bro, I kind of miss Massachusetts. The thing about this gel, I think I probably want the trial one because, all right, when we get to so we'll have to figure out which stain to get. That's still on the list, but let's go back to the reading this because I didn't read all this. Okay, so we have this, I don't know, a bunch of just random liquids, man. Wait, what? Wait, I don't understand this. Okay, finish PCR. To the PCR tube, add 10x cut smart buffer. Okay, then okay, half a microliter of this stuff. And how much do you get? And what's the okay? So now let's look up. Okay, so here's the product spec for the ladder. So safe 1 kb ladder. And we have the storage is at 4C 24 month. The volume is does it tell me the volume? I don't think it tells me the volume. Because I guess it can be varied. But you buy 1.25 milliliters. And the shelf life 24 months. And then I guess we could store the concentrate. We could store all of this stuff. I guess they don't give you this SKU. That's fine. So then we should just store some sort of catalog number, which makes sense. So it's NEB. Okay, and then we need the cost too. The cost is something that they don't like to talk about because it's so expensive, but it's $87. Okay, now we're looking at suitable for 125 to 250. So you're supposed to add like 10 microliters to this thing? Because this is 1250 microliters. So all right, so let's just move on to the next one. The next one is going to be the DNPI, I guess. All right, so the DNPI, NEB. Let's go to the product specification. Okay, so this is going to be $77. Quality, safety, and legal specifications. The specification sheet is a document that includes all that I need. Okay, good. Okay, so this needs to be stored at minus 20C, and its shelf life is 24 months. So I'm willing to pay that. So we can move on to the next part of it, which is this R Cut Smart stuff. Okay, so R Cut Smart. Wait, what? Okay. Okay, let's go to the product spec. This is, what did it say? $27? $29. Okay, minus 20. 36 months. And it's going to be 5 milliliters. Okay, we need our polymerase too. Okay, let's get the Monarch thing. Well, actually, we want to look up if we get the Monarch. PCR cleanup kit. What is this? Exosip? Yeah, this reminds me a lot of the LCSC. You're just looking through specs and just trying to figure out what you need. It's procurement, man. Procurement. Okay, so this is... All right. I mean, basically, like 6 months is just a little bit too little for me. Okay, but we need to go through. So we did the BZ. And then the EY doesn't have its own, but maybe this manual, maybe. But, oh, here we go. We got the safety sheets. But so if we look up a composite product, does it give me shelf lives? Okay, so you can just store them room temp. All right, so that's cool. So all the stuff in the Monarch cleanup. So we'll say 50 reactions. It's 24 months, 25C, 24 months. And it's how much? It's not that much. I don't think it's that much. It's $102. I mean, we're going to get all this information and then figure out what actually, you know. Okay, so wait, let's review. We have liquid culture. We're going to mini prep it. I have all the mini prep stuff, so we're good there. We're going to dilute down the result of the mini prep, store the rest. How do you store plasmid? What's the long-term, the recommended long-term storage of plasmid 19? You know, there's a lot of myths here, man. It's like, yeah, but does it really matter? Like, the problem with this is that it's overly safe. It's too safe. Okay, so we're on step. Okay, so wait, let me just write out the procedure again. Okay, the plan. Okay, step 1, liquid culture of E. coli expressing PUC19. Mini prep to purify the PUC19. 3, quantify concentration on nanodrop. Dilute some for PCR. Maybe store the rest in TE stock. -20. Oh yeah. Okay. PCR. Run some of the PCR product on a gel. Okay, so now we do a DPN. And then after we've cleaned it up, we could probably rerun it on a okay, Okay, so we've done the PCR cleanup. Now it's time to. So this is basically the plasmid linearization and preparation. Okay, and then now here we want the fragment preparation. And we need to do a bunch of calculations because I don't. Because there's all those guidelines for the actual assembly that I don't remember if they want. Because they want like 50 nanograms of vector and 100 nanograms of. I don't remember. Oh no, I think it's a 1 to 2 molar ratio. Let me take a walk around the block. Okay, we're gonna end the stream here because the BioCurious board meeting is happening now, and I am going to go there. Well, I'm gonna join the call. Um, I think genuinely tomorrow might be the day that this stuff gets ordered and I actually do this thing. It's pretty incredible that it's not only taking this long, but thinking back, like where I started and where I am now. The primer design, you know, the tools. I'm totally comfortable with Snapgene, Benchling, that thing that any builder, you know, the general protocol is clear to me. And I guess the thing is, if I want to, I guess, okay, so wait, hold on. I thought I had that. Or hi-fi. So this is the most expensive thing. I don't know why this is so expensive, but so I'm currently at $500 about, which would make this by far the most expensive, and we're not even done. I mean, I'll probably spend $1,000 on reagents. By far the most expensive project I've ever done. And it's like Bio 101 stuff. That's, I guess, the thing. That's crazy. Okay, but if I just get the hi-fi a master mix, how much cheaper is this? It's not even that much cheaper. That's the thing. Dude, see, if I had access to unlimited reagents and a cloud lab, bro, I'd be running crazy experiments. I'd be generating like, could I do, like 100 fragment assembly? When does it stop working? You know, with the repeatability and no pipetting, you would actually get clean data and just see the results, and it'd be so interesting. Okay, wait, we should look at the spec here because, probably this one, right? Okay, so I think I should just buy some of this stuff, right? Ladder, um, we need the, so the kit is -20, and it's for 10 reactions, which is crazy to me. I mean, 10 reactions? This hi-fi stuff is expensive. And it's 18 months. Do they have an NEB student discount? No, that's dumb, man. Oh man, that's fun stuff. Love getting to know my sales rep. I love getting to know my sales rep, bro. I love getting to know my sales rep, bro. All right, let's close some of these specs because we have most of the data. Um, yeah, this thing sucks though. I guess we need to figure out this. Um, okay, all right. I'm gonna end it now. So there's still basically the agarose, the I guess the loading die. Well, the polymerase, basically loading die, polymerase, the buffers, TAE buffer, the DNA stain. So we have 1, 2, 3, 4. We have 5 things. Okay, so all right. So, it's like why is it So why is it so much? Where can I get free stuff from? Free samples, Thermo Fisher. Free samples. Is this what master is this a qPCR? I don't need qPCR. But I want free stuff. How do I earn points? You earn points by spending money. Um